are typically divided to subregions called domains. For instance, if we look at the protein hemagglutinin, so it has two domains. It has a region that's packed and has a ball shape, so a globe shape, so we call it the globular domain, the gray color. And then down here, the light brown has a cylinder-shaped structure, so we call it the fibrous domain. And that, to put aside the fact that it's built of several units, it's a multimeric unit, it's a multimeric protein, but each unit has two distinct domains. Okay, so might not be the most well-defined explanation, but structurally a domain is a compactly folded region of polypeptide. It's what biologists look and say this looks like a ball, so we go globular and so on. Okay, and like motifs of secondary structures, domains are incorporated as modules into different proteins. So a module here means a structure, a well-defined, relatively small structure that you can use in other proteins, something like a piece of Lego that can be used for a certain purpose and for another one and so on. For instance, EGF is the name of a protein. It's a monomeric protein, and it's built of a single domain, a globular domain, but its domain can also be found in another important neurobiology protein that's called neuroligin. So the same domain is found here in addition to other domains. And it can be found in several copies in the EGF precursor protein and so on. So a domain by biologists is sometimes referred to as a module. And same thing for the secondary structures, the protein motifs that we mentioned. And the idea is that we think that once nature found a structure that's good for some purpose, it's strong enough to be a structurally stabilizing structure, or it interacts very well with calcium ions and so on. So it's being used for other purposes or other locations in the cell by several proteins. So that's the idea of modules in this context. And lastly, the fourth, and as I said in the beginning, including the sometimes fifth structural organization, the quaternary structure. So some proteins are not just a single unit, but are composed of several units, and sometimes are even composites or assemblies of several proteins. And not only that, but sometimes you find big, what they call machineries that are composed of proteins and also nucleic acids embedded in them. For instance, the mRNA transcription initiation machinery. So at this level, again, it's usually called a complex or a machinery if it does some well-defined task. So for instance, in this machinery, we have the RNA polymerase, that's a protein, the general transcription factor is another bunch of proteins. We have several RNAs embedded in it, and a not very specific name, a mediator complex for really another bunch of proteins that glue the whole thing together. So that's a complex or a machine. And if I will tell you what are the subunits that compose this machine and the ratios, the stoichiometry between these units, that would be the quaternary structure description. All right, so these are the four levels. Okay, a word about protein evolution, just a word. So as we said at the beginning, there is a relation between the amino acid sequence and the spatial organization of the structure. Now we can calculate similarity between sequences, and we can also calculate similarity between spatial organization of proteins. Just put them both together and measure the distance between the atoms, which are presumably the same atoms that play the same role in these two compared proteins, and then, I don't know, average across the whole distance. And there are different measurements for doing so, but all in all, if you prepare an evolutionary tree or a similarity tree between the sequences of proteins and between the spatial structure, you would, all in all, come to the same conclusion. The trees, the evolutionary trees, will look 90% the same. Okay, another term, homologs. So proteins that have a common ancestor are referred to as homologs. Let's see an example to explain this idea. So we are looking here at the hemoglobin. Hemoglobin, that's the protein that carries oxygen to the tissues in mammalians. It's built of 4 units. It's a tetrameric protein. But look how similar just the beta subunit of this protein to the more, so this is the beta subunit of the hemoglobin we have in our body. And these are more ancient, this is a much more ancient lingohemoglobin species or molecules perform the same task. And you can appreciate the similarity between the two. You can again calculate the similarity and compare it to similarities with the rest of the proteomics, with the rest of the proteins that exist in nature. And there's a high degree of similarity. This is the hem in red. That's not essential. Okay, so these two proteins, the hemoglobin and myoglobin, for instance, are homologs. Or the hemoglobin and myoglobin are homologs because they are derived, so it's not from a common ancestor. Right. Okay, so we met the 4 structural level of organization in proteins, and we mentioned protein motifs and protein domains and lots of relating concepts. We will now proceed with the modifications that proteins undergo after they are being synthesized. So they are being folded, actively folded, they are being modified chemically, and eventually they are degraded, again in an active process that we'll describe. And let's start with modification, with folding. So in vitro, in the test tube, a nice calculation of how much conformations are there for a single polypeptide would be that any polypeptide chain that contains n residues can in principle fold into 8 in the power of n, simply because there are n, for chemical reasons, there are n angles that are allowed in a polypeptide, or stereochemically allowed. And so you reach with very relatively short polypeptide, you reach an enormous amount of possible conformations. But in general, even in vitro, not only in cells, excluding any factor that actively helps the folding of protein, in general, all molecules of a protein species adopt a single conformation, which is called the native state. And most usually, or for the vast majority of proteins, the native state is the most stable form of the molecule. So if we will plot an energy, the energy of each, or the energy, this function of the conformations that exist for the protein, for F protein. So again, so at the native state, you would usually find the native state in the global minimum, and sometimes in the local minimum, or in a local minimum. Okay, but again, for the vast majority of proteins, this will be the, so these are conformations. Should be not a single x-axis, should be a multi-dimensional plane, but never mind, but just if you look at the energy in the different conformations that exist. Okay, we'll go back to that in a minute. Alright, so now we're going into the cell. So in the living cell, in vivo, the folding of a protein is promoted by molecules called chaperones. And there are 2 kinds of chaperones. The first are called molecular chaperones. These ones bind and stabilize unfolded or partly folded proteins. and they prevent aggregation and degradation. If there is an improper folding of a protein, usually downstream you will see aggregation and eventually degradation of the protein. And needless to say, it will be unfunctional. The other kind are called chaperonins, and they are directly facilitating the folding of proteins. And the next slide will show you the distinct mechanism of action of these two. Okay, so for some proteins, as they are being synthesized from the ribosome, described as the two gray spheres, this is the machine that synthesizes proteins, link one amino acid to another, to be right to the polypeptide chains. So for some proteins, which are described here as the green tail, molecular chaperones, the yellow spheres, bind them as they are synthesized, inhibit some local foldings that shouldn't occur, and promote several other foldings that are the proper foldings. And for some proteins, this is enough to achieve the correctly or the properly folded form. For other proteins, the molecular chaperones again inhibit local improper folding, and in parallel to that, guide the proteins to a big protein complex that's called the chaperonin. So these are chaperonins. The folded protein is inserted into this cup-shaped huge complex, and then even a seal is being added. A cap is sealed on top of the cup-shaped chaperonin. It's like a magic box, and then inside the magic occurs, and the protein comes out properly folded. Now there are some nice computational models of what happens inside chaperonin. So I'd like to show the principal conclusion of this model. So outside, without the chaperonin, each protein, as it is being folded, or as it is being on its way to the global or to the native conformation, it passes through several other conformations. So if we draw some conceptual trajectory of that the protein performs in the conformation space, it will look something like that. So again, it's not really a plane, but rather a multidimensional plane, but these are the plane of all possible conformations. And the protein, so this will be the start side. And the protein, well, if you assume a random process, the protein probes this conformational space until it reaches its ending point, which is the native state. So this is in the absence of chaperonins. What chaperonins do, what happens inside this magic box, or so it is thought, so they just inhibit or prevent some regions, some conformations from being possible. So for instance, all this region is now wiped out, and also this one. And if you still consider a random process, so the protein will have a much shorter plane to probe, and it will reach faster the ending point or the native state. So it will happen much faster, and this is the way they promote the correct folding. Because again, in vitro, without chaperonins, proteins will achieve the proper fold as well. So what do? They promote, or they make it faster simply by doing this. It is actually the same thing for the molecular chaperones. Because again, they just inhibit specific foldings from occurring, and so you're left with a smaller space of conformations that you need to probe on your way to the native state. Okay, let's just mention this. Molecular chaperones consist of the HSP70 and its homologs. So this is a very important protein that is a molecular chaperone. It was historically discovered first as having this feature, and so we call it HSP and its homologs. Okay. So there's a nice movie, but for now it doesn't work, so perhaps we'll see it folding in chaperones and chaperones. Okay, so that was protein folding. Let's proceed with modification. Okay, it's a very frequent event. Nearly every protein in a cell is chemically modified after it is synthesized on the ribosome. Modifications can be a linkage of a chemical group, either to the N-terminal of the protein, which you've already defined, the N-terminal, or somewhere in the middle of the protein. And these modifications alter everything that the protein performs. They alter its activity, the lifespan, or even the location in the cell. Okay, let's see the first modification. So we're looking at acetylated N-terminus of a protein. Adding an acetyl is called acetylation. Acetylation can actually occur also in internal region of the protein, but here we are looking at acetylation of the N-terminal residue. So this is the N-terminal of a protein that sits right here. This is its N-terminal, and this is the red is the acetyl. It's being added on these N-terminal, and well, it was shown that non-acetylated proteins are rapidly degraded by intracellular proteases. So proteases are proteins that degrade other proteins. Intracellular because there are some proteases that are located outside of the cell, extracellularly. And so again, if you don't have this protecting acetylation, you'll be rapidly degraded by proteases. So again, 99% of proteins have to be acetylated after their synthesis. Okay, in a bird's view, here are some other existing modifications. Modification on internal residues, which again affect biological processes, location in the cell, and so on. So glycosylation is the name of linking a saccharide to protein. It again is thought to be a protective modification because we know that a lot of secreted proteins are glycosylated, and it's thought that it's to guard them from the harsh environments that are out there outside the cell. So again, what we do know is that, again, secreted proteins are glycosylated. Acetylation could be on the N-terminal as we saw previously, but it also could be on a residual group somewhere in the internal part of the protein. So for instance, this is an acetylated lysine. So it is now called acetyl-lysine, amino acid lysine. Okay, phosphorylation, the addition of a phosphate on some internal residue. Hydroxylation, the addition of a hydroxyl. Methylation, the addition of a methyl. Carboxylation, the addition of a carboxyl. Okay, we mentioned that there are 20 amino acids, right, from which polypeptides are built from. But actually, when you take into account all possible modifications, there are actually effectively much more than 20 amino acids. Because again, you can have acetyl-lysine, phosphorylation, and so on. So this is just a note of caution when you try to simplify things. Okay, modifications are not only an addition of a chemical group to the polypeptide, but there could also be processing in the sense of taking out fragments of the protein. Okay, so peptide segments of some proteins are removed after synthesis. An exemplary processes in which this could be found are blood coagulation, digestion, and programmed cell death. So these are all processes that have to be in which a rapid response is involved. So if you already have a protein synthesized in the cell, then what you need to do for it to become functional is to remove some segment from it, and you don't need to synthesize new genes or translocate these proteins to the right location and so on. Then the response is much faster. So for instance, in blood coagulation, when we are injured, some protein segment of a specific protein is being removed, and that starts initiates a cascade of events in which the blood starts to coagulate. So again, the response is much quicker when the mechanism is processing or removal of segments from existing proteins. It's like a sword inside a sheath, and you only have to take up, and the sword and the sheath are already there in the cell, are already synthesized, and all you have to do for the sword to become functional is to take off the sheath, and then it's a much faster mechanism. Okay, it was not too late ago was thought that degradation was a passive process for proteins, but we know that it's a very active and regulated process today. Okay, we should start by saying that the activity of a protein depends on its amount in the cell, which in its turn depends on its rate of synthesis, right? But also its rate of degradation. And maybe a decade ago, a bit more than that, the degradation was always a scalar, was always something stable, so researchers thought. And now we know that it's an active, and for different proteins there are different rates of degradation, not only different rates of synthesis. The lifespan of proteins varies from as short as a few minutes for mitotic cyclins, in which the very accurate temporal resolution should be very should have a high temporal resolution. To the most extreme example of protein of the lifespan of a protein is proteins that are at the same age of the organism, proteins that reside in the lens of the eye. So these are proteins that do not degrade ever, and we are born with them and die with them.
The rate of degradation is related to the rate we discussed in the first lecture.
The rate of what's the rate of synthesis?
There's something that balances synthesis and degradation of some substance.
No, so this was the chemical equilibrium.
Yeah.
So no, not really. You can maybe try to put the chemical equilibrium concept on this thing, but not really. No. Chemical equilibrium is from a chemical reaction. It's when the product is at the rate of product production stable or equalize the rate of the reverse reaction of reactant production. That's called chemical equilibrium. So it's a different thing here. Yeah. Okay, degradation of extracellular proteins takes place inside inside organelles that are called lysosomes. We will see the structure later on, but I just wanted to mention it here. So when you go back to this slide, you link these lysosomes here, and this is in opposed to so lysosomes, just to say a word, are membrane-limited organelles having an acidic interior, an interior that's good for breaking down proteins. So this is true for extracellular proteins. And for intracellular proteins, cytosolic proteins, the cytosol is everything that's inside the cell, excluding the nucleus, or other organelles that are there, which we will meet. So for cytosolic proteins, the degradation occurs via the process of ubiquitination, which is actually a chemical modification, another chemical modification from the bunch of modifications we've already introduced. And here we're talking about a modification that happens on a lysine side chain, and the modification itself is adding a molecule that's called ubiquitin. This is a 76 residue polypeptide, a small polypeptide, and when you add enough ubiquitin, that leads to degradation of what we call the ubiquitin-tagged protein. The degradation of these proteins occur in proteasomes, another complex or spot in the cell which we will meet. Okay, now this process and the characteristics of it were discovered by two Israeli scientists, Hershko and Chekhanouver, who won the Nobel Prize for it in the house. So we will now very briefly explain the mechanism of ubiquitination. So ubiquitination starts with two preliminary steps. Preliminary because the green there is the target protein, the protein to be ubiquitinated and then degraded. So there are two preliminary steps beforehand. And these are loading of the ubiquitin UB molecule on an E1, which then promotes its loading on the E2, which then promotes its loading the ubiquitin on the. So in the third step is when the ubiquitin, the first one, is added upon the target protein, and then the same thing happens a couple of times to give rise to several ubiquitin molecules attached to the target protein. And when the protein looks like this, then it is recognized by the cell to be guided to the proteasome, the protein slaughter machine, which degrades everything that comes near it. And the outcome is a degraded peptide. Okay, maybe that would be a good point to say a word about, well, the exam is very far away, but not the exam, but just how deep or what details do you need to remember for each slide? An interesting question. So, okay, there's no clear line I can draw that this will be in the exam, or this you should remember, or this detail you should not. But our agenda is that the principles of the story are what matters most, and the examples and the E1, for instance, E2, I don't know, third reaction and so on, or third step in the reaction, are more the details that serve to consolidate better the principles themselves. So again, I don't want to guarantee on any this detail or that. The more the better, obviously, but I could safely say, for instance, from this side, you do not have to remember that E1 loads the ubiquitin, the E2 loads the ubiquitin, but all in all that we're talking about a cascade of loading between several, I don't know, enzymes or proteins, which end with the several molecules of ubiquitin tagged to the, and so on. So, the guidelines of the principal lines of the story are the ones that we should take with us. Make sense? Is that okay? Because I was already asked about that. That's a good question, but again, I don't want to draw a red line. Okay. So that was the gradation. Again, we're going to skip the movie because there is no audio, but I'll fix it for a second. Okay, we will now start with describing a bunch of proteins that promote chemical reactions. These are enzymes. Okay, enzymes promote a chemical alteration of a molecule that they recognize, and these molecules are called ligands or substrates. In the context of enzymes, they are called substrates. So enzyme acts upon a substrate and promotes its alteration to something else. So again, we're looking here, as we did last time, on an energy scale of an exothermic reaction, in which the reactants are in higher energetical level than the products. And I think I mentioned it just briefly last time. So instead of having a monotonously decreasing curve from the reactants to the products, we actually have what is called activation energy, a threshold we need to pass in order to slide down towards the product side. And what the enzymes really do is decreasing this threshold. So blue is without an enzyme, and red is in the presence of an enzyme. They do so by stabilizing the intermediate species of the molecule, the intermediate state of the molecule that's right here, and thereby decreasing the activation energy and allowing the reaction to occur much faster. Okay, several features of enzymes. They have immense catalytic power. They can promote or increase the rate of a chemical reaction in thousands folds. And they have high degree of specificity. So it's enough for you to change some 2 or 3 atoms in the substrate, and it will no longer be recognized as substrate for the enzyme. So they are specific to a certain substrate. Certain amino acid amino acid side chains of an enzyme form the active site. So this is the site where everything happens in the enzyme, and it's subdivided to two regions. The first one is the binding site. So the active site is subdivided to two regions. In the active site, there is the binding site that binds the substrate, and the catalytic site, where catalysis occurs. Okay, active site subdivided to binding and catalytic site. Alright. Here's an example of an enzyme. It's called protein kinase A, or just PKA. This enzyme adds a phosphate group on certain amino acids. So we mentioned the chemical modification of phosphorylation. We will later on see that it has to do with regulation of proteins, or their regulation of their function. And this is an enzyme that performs this chemical modification. Okay, so the gray spheres, or the water accessible surface model painted gray, is the pKa. It has two distinct domains. Again, just for using the terms that we defined, it has a small domain and a large domain. The red trace bond, the red, how do you call it, the carbon trace model here is the target peptide on which a phosphate is going to be added. And the green here is the nucleotide from which a phosphate is going to be removed and added again on the target peptide. And the blue region in the pKa is what we call the glycine lead, and that's a region that, so we mentioned that the tertiary structure of the protein is continuously fluctuating, and once the pKa meets both the target peptide and the nucleotide, and these two molecules are inserted into this region, so it undergoes induced fit. The tertiary structure stops vibrating, and the glycine lead locks upon these two molecules and holds them in the correct orientation in relation to one another, and in very close proximity. Okay, so in this pKa we saw the substrate-induced fit. It's substrate-induced because once it meets its substrates, then you have the fit. And okay, we just mentioned that there are several other protein kinases which differ between them mainly in the region that's below the target peptide. So this is the region that will recognize different segments of peptide. So pKa will recognize the red peptide as a substrate, while another pKc will recognize a yellow peptide, target peptide as a target, simply because of different amino acids below the target peptide, or in the target peptide binding site. Alright? So we will now focus on the active site, pKa. The next slide will be a zoom in to this region. Yes.
This all happens in the large domain?
The active site, I think, is right in the middle of the two domains, but I'm not sure if it's located just on the large. Okay, so again, we are zooming in to the active site right now. Okay, so let's start with everything that is not the pKa. The point will be there. Okay, so the black here is the ATP, is the nucleotide with its first, second phosphate, and the third phosphate that's going to be taken off the ATP to give rise to ADP, and it will be linked to the blue target protein, or target peptide. So here, and outside the figure, you have a peptide, that's the target peptide, and what you see here is only the serine residue of this target peptide, on which the phosphate taken from the ATP is going to be linked. Alright. And everything else, these amino acids and the charges that are distributed here, is the active site of the pKa. And now, these positive charges here induce what is called electron, where is it? Electron delocalization. Okay, these positive charges right here induce electron delocalization from this third phosphate group, and that this causes the bond that's about to be broken. It's shaking this bond from between the second and third phosphate, and it makes the bond that's about to be formed possible. The bond between the phosphate and the target, and the serine from the target peptide, serine from the target peptide. Okay, and this is exactly the intermediate species, the intermediate state. This is the species that stands in the maximum point of the activation energy in the chemical reaction that's promoted by the enzyme, pKa. So the active site of pKa is designed to stabilize this species, this state, the intermediate state. Okay, now two things can happen from here. Either you slide down towards the product, in which case, so this is the products, right? The ADP and the phosphorylated serine. So either you slide down towards the product, in which case the glycine lead opens up, and the ADP and phosphorylated target peptide dissociates from the active site. But also you can slide back towards the initial state, in which case the glycine lead stays locked and still holds the two substrates, the ATP and target peptide, close together. And again, you're pushed towards the intermediate state. Okay, so taking these two principles together, the first one is that the pKa stabilizes the intermediate state, this state in which you have delocalizations of the electrons around the third phosphate group. And the second principle I described, in which if you slide, if in this state the glycine lead opens, but in this case it stays locked. Taking these two principles together, you get a huge promotion or increase in the rate of this reaction. A reaction that says ATP plus target protein equals ADP gives ADP plus phosphorylated target protein. Okay, so now again, the delocalization is less essential, but here the two principles we just mentioned are really at the base of the pKa mechanism of action, of what happens in its active site. Yeah. Okay, so that was the atomic level description of what happens in pKa active site. Okay, enjoyed it, and okay, a few more words about enzymes. So in order to improve reaction efficiency for reactions that are coupled, you see most often in cells that enzymes are assembled on a common scaffold, like in the pyruvate hydrogenase, multimeric enzyme. So again, if it's a complex or structural complex, we usually call it a protein complex, and here we call it a multimeric enzyme, or yeah, usually I don't see enzymatic complex, but never mind. Multimeric enzyme, meaning that it's built of several enzymes that promote chemical reactions that are coupled together. So this is the net reaction that the pyruvate hydrogenase perform, but it's made of several smaller chemical reactions that promoted each one by another enzyme. These different reactions balance one another. Okay, when this is the case, so the products of the first chemical reaction are immediately introduced as reactants for the second in the line for the next reaction. Right, so that's what gives it the multimeric enzymes their higher efficiency. So we introduced, so that's it for today. We introduced the hierarchical structure of proteins. We discussed folding, modification, and degradation by ubiquitination of cytosolic proteins, and described the basics of enzyme. We will proceed next time with another group of proteins found in the cell, this time proteins that are responsible for molecular motility, for movement in the cell, and then proceed with the other, with the rest of the chapter. Please answer the question page, which I will also send by email and which is also in the website for the biochemistry chapter we ended at the beginning of the class. And again, and if you'd like to, I'll already send you by email the article that we will read together at the end of this chapter. So for those of you who already want to have a look at it. Any questions? Okay, so see you next week.
Thank you.
לא, לא, בעצם היה באימייל אחר. שלך זה... גם עם שלך. נראה לך בעבודה שלך לדבר? כן. לא, אפילו כתבתי את המייל, ואז זה יהיה פסיכי. ואז השאיר... לא, אבל זאת ידעתי כאילו את המייל הוא לא הכניס, את הכתובת משום מה. לא, ראיתי את זה, קצת פנימי. קצת פרט, אני כאילו...
כמה פעמים יש בפיקייטר?
Oh, good. I wasn't here Thursday.
Right.
Sorry I couldn't make it, but you mentioned something about the homework. I don't think the homework.
No, okay.
Yeah, I don't think is it on Moodle?
So it's on Moodle. You have it there, and you know, 90%. So, but anyway, what you saying by email? Did you write or anyone else wrote your email address?
Yeah, no, I think
So
I think
if
you
you
don't have to.
so if you can just put it here, and I'll add you on the group. This is first thing, and second is so yeah, it's on Moodle also. This year is Moodle.
Okay, perfect.
And
I'll look at the Moodle, but yeah, it'd be good to be on.
I'll add you on the group and then send it by email. And it's really not. I mean, it's not for submission. You just answer it yourself alone or your friends and then you will get it.
Okay, good.
Because you went there.
Yeah.
Okay.
Thank you.
חוברת, זה חוברת של לא, בקיצור לא. אבל יש טקסטבוק, לא, יש טקסט, אני לא מתכוון, זה ספר, אז זה יש אפילו PDF. אני לא יכול לשים את זה באתר, כי אז יתבעו מישהו פה. יש לך לדיסק קומפיוטר? יש לי. לא. כן, כבר עכשיו התקיימו את השם של הקורס והכל, אז אמור להיות במסמך.
shallowly, if I can say so. Just the general idea. The motivation for studying this is that many times you can look at a neuron or even in general a cell. You can look at the membrane of the cell as having electrical properties, such as capacitance and resistance, and this is what we're going to go over. The second topic is completely different, Euler's numeric integration, order 1. It will probably be in the third class. So, RC circuit. Okay, so a few basic concepts that you have to be familiar with. So the first thing we talk about when we talk about electricity is electric charge. Electric charge is a basic fundamental property of a particle, of any particle. A particle may or may not have charge. It may have positive charge or negative charge. You may already know that positive charges, two identical charges, identical sign charges are repelling, and two opposite sign charges are attracting to each other. So charge has to do with forces. Okay? So charge is measured in Coulomb. Okay, this is the unit, the physical unit used to measure a charge. And we're not going to write this every time. We're just usually just going to use the letter C or sometimes maybe more. And the letter that is usually used to measure charge is Q. Okay. Symbol. Of course, you can use any symbol. It's your decision. Q. Okay. When we now know what is charge, so we can talk about electric current. Okay? So when charges move from place to place, usually they have to move within some material which is conducting, so it allows their movement. So when charges move from place to place, they generate current. It's just the movement of charges. So we usually use the letter I, and it's measured in ampere. Okay. Now there is a mathematical relationship between the charge and the current. In fact, if you think of it, then let's say that I have a conducting material, okay, some kind of a wire, okay, a metal wire which is conducting, such that charges can flow through this material. So let's say that I have a Q amount of charge, okay, 1 Coulomb of charge, and it's flowing from side to side. So the definition of current is how much charge is going through one section of this material in 1 second. Okay? So if, for example, I'm looking at this one section, I'm taking the wire and I'm just looking at this one section, and I'm asking how much charge went through this section in 1 second, and the answer is 1 Coulomb. So the current through this section is 1 ampere. Okay? So basically what I just told you is that the change in charge is the current. Okay? Now the current can be the amount of charge that is flowing through this section doesn't have to be constant in time. It can be variable. This is why we can't just use this measurement. We have to use something that is more precise, and that's a derivative. Okay? So the change in the charge amount is the current. It can be a function of time if this derivative is a function of time. Okay, the third concept is voltage, electric voltage. So voltage is more of a relative concept. We use the symbol V, and the units are volts. So we also use V for the units. Okay. And so why is it a relative concept? First, before we define voltage, we have to say that in every point of space we have what we call electrical potential. Potential is what is caused by the fact that there are electrical charges in many places. And we know that charges either repel or attract each other. So if I know that there is a charge here, a charge there, and I know their size, then in this point of space, there will be some force acting on a new charge that I'm bringing. Okay? So this point of space has some potential. Okay? It's measured as a potential. Now if you take 2 points in space, and you calculate the difference in their potential, this is the voltage between these 2 points. Okay, so voltage is between 2 points in space. Okay, it's a relative concept. Okay. Now, what does it mean? Let's talk about the relationship between current and voltage in general, not the mathematical relationship yet. For example, if I have a positive charge, and there is a path from the place where it is and another place, and there is a voltage difference that's between these 2 points, then the positive charge will always flow from the positive voltage to the negative voltage. Okay, from the high voltage to the low voltage. Okay. So if I have 2 points, and this one has a higher voltage than this one, and there is a wire between them, then if I have a positive charge, then this will be the direction of flow. Okay, positive charge. Okay, a negative charge would flow to the left. Okay, so it is repelled from the negative voltage and attracted to the higher voltage. Okay, next now that we know all this, we can talk about electrical components. Components are the building blocks that we use in order to build circuits, electrical circuits. I'm going to introduce you 2 passive electrical components, a resistor and capacitor. So let's try to see how we define them. Okay, so the first one would be a resistor. Okay. This is the symbol of the resistor in the electrical circuit. Okay, so it has 2 terminals, 2 ends. Okay, we call them terminals. So it has to connect to the electrical circuit in 2 points. If it's only 1 point, then it's not connected to the circuit. Okay. This. Okay. What is physically what a resistor is? If you take some conducting material, but you insert some sort of disturbance in this material, so it's conducting, but it's not conducting perfectly. So particles cannot flow with the speed of light within this material. So they have some obstacle that they have to go through. So this is resistance. Okay, this is what this component is doing. Okay, so it has a resistance, measure it, the resistance, the symbol is usually R, and we measure it in ohm. Okay. And the most prominent way to describe a resistor is that is the relationship between the voltage over it and the current through it. Okay, so let's first mark the voltage over it. Let's say that the voltage over the resistor is from here to here. Okay, such that this is the plus and this is the minus. I can either write plus minus or use this arrow from the minus to the plus to indicate that the voltage is in this direction. Okay, positive voltage in this direction. So this is the voltage. It has, I don't know, 1 volt, 5 volt, whatever, some amount of voltage over it between the two terminals. And there is some current through it if it's connected to some circuit. Okay, let's say that it's connected to some circuit and there is some current flowing through it. So the arrow that's indicating the current is through the resistor, not between the terminals. Okay, through it. Okay. So you've probably heard of Ohm's law, which is the relationship between the voltage and the current of the resistor. It's linear, and the coefficient is exactly the resistance. Okay, this is how we define resistance. The resistance is defined as the ratio between V and I, and it's always constant, no matter what V is or what I is. If I change it, if I change I, then V will be changed such that R is constant. Okay, so let's try to see if this makes sense to us. For example, if I force a high voltage, if I force some voltage between the two terminals, then I get some current through the resistor, such that this formula is correct. If I increase the voltage, what happens to the current?
Goes up.
It also increases by the same ratio, right? If I increase the voltage twice by 2, then the current will also increase by 2. And this makes sense because when I increase the voltage, then there is more repellence in these points of positive charges. So positive charges tend to go through this obstacle even faster. And more current, more charge is flowing through an intersection of this resistor in 1 second. Okay? Of course, note that V and I can be functions of time, but they are always proportional to one another, with a proportion R. Okay, sometimes we use, instead of R, which is resistance, nagdut, sometimes we use the inverse. Conductance. Molichut. Okay, so if we want to use the terminology of G, then the relationship is simply I is equal to VG. Okay, it's exactly the same formula. Right? So conductance is 1 over the resistance. If the resistance is high, then it means that it's very hard to go through the resistor, and the conductance is low. Okay? Okay, let's take two, just as an example, let's take 2 resistors. One of them is big and one of them is small. Okay? 1 kilo ohm, 1,000 ohm, and R2 will be 5 kilo ohm. Okay? Now let's try to see what happens when we force a voltage of, I don't know, 5 volts over both of them. Okay? They're not connected. We're separated. Okay? What is the current through each one of them, in each one of the different cases, when I apply a voltage of 5 volts? Which one has a higher current? And does it make sense?
The first one, 5 milliamperes, is 1 milliampere.
Yes, okay. So naturally, when the resistance is higher, then the current has to be lower to get the same voltage. Does this make sense? Yes, because when the resistance is higher, then less charges can flow by. Okay, so that's the idea. Oh yeah, and conductance is measured in Siemens. I've also, what? No, I've also seen this mark. Just the opposite of it, but more. Okay. So the second component that I'm introducing is capacitor. And now you know why I call this lesson RC circuit. We only have two types of components, resistors and capacitors. There's also another useful passive type, which is an inductor, SNIL, but we're not going to study it because then, in order to solve the circuit, we need differential equations of second order, and we only know how to solve first order differential equations. Okay, so capacitor, this is the symbol. Physically, again, it has two terminals. Okay, it has to be connected in two spots. Physically, a capacitor is simply two parallel plates of metal that are separated by an insulator, a material that doesn't conduct electricity. So current cannot flow between these two plates. But the rule is that current is being accumulated on those plates, not current, charge, I'm sorry, such that the amount of charge that is accumulated on one of the, let's say that I have Q charge on the left plate in this case, and it's a positive charge, is exactly like the amount of charge that is accumulated on the second plate, but with a different sign. So I have minus Q here, opposite sign. Okay, so at any moment of time, if you measure the amount of charge that you have in the two plates, it will be the same amount with opposite signs. So what happens if is that if I connect this to some circuit and current is flowing through, I just said that current cannot flow through here, but basically I will get a picture as if the same current is flowing here and here, because if I have 5 coulombs entering this plate, then I have to have 5 coulombs leaving this plate. Okay, plus 5 coulombs in these two cases. Okay, so I will get something that seems like a continuous current, and indeed, whenever you connect an ideal capacitor into a circuit, you'll get a continuous current. Okay, you can always assume that it's like here. Here I didn't even bother to say that it's the same current here and here. Okay. Okay, so this is a capacitor. Now, it has a capacitance, Kibul. We use the symbol C. We measure it in farad. And the formula that connects, we have a formula that's connecting the amount of charge that is on each one of the plates with the capacitance and the voltage. So this is the formula. Q is equal to VC. So at any point of time, for a given capacitor with capacitance C, if you know the voltage over it, then you know the amount of charge that is on each of the plates. One of them is plus, one of them is minus, but you know the amount. Okay? Or if you know the charge and you know the capacitance, then you know the voltage that is between the two terminals of this component. Okay? And since I know this formula, from this formula I can derive another formula, which will be the most important thing, the most useful for us formula for each of the components is the relationship between I and V, the VI relationship. So here it's linear. Okay, it's very easy. Here I don't yet have the relationship between V and I, but I can use my previous knowledge in order to derive it. Okay? So I want to combine these two formulas in order to get the relationship between I and V. You already see that there will be a derivative, but it doesn't matter. It's still the relationship. So who knows the relationship between I and V for a capacitor? I is equal to dq maybe, copying. And I know Q. I know the formula for Q. I just have to differentiate this V times C. Now, I'm assuming always that in our components there are ideals such that R and C are constant. We don't play with them in time. So R is C is a constant. So when I differentiate, I just copy. V may or may not be a constant. It could be a function of time. If someone is enforcing a variable voltage, they can do it. Okay? Yes.
I had a question. Why are we using V instead of T here? Just I know that
Instead of?
כאילו כאן עשית את זה עם V, באמת, אני מכיר בהגדרה של I
You can ask in English because the people
Sorry, that I know the definition of I being as in the movement of charges over time, so why are we doing it here as in the
Sorry?
difference
Yeah,
in the...
I just miscopied. Thank you.
Okay.
Okay, so I have to differentiate this by time. So, I don't know. Depending on what V is, the derivative of V with respect to time. Okay? So, here we have the relationship between I and V for a capacitor, for an ideal capacitor. Okay? What does this mean, this relationship between I and V? Let's try to get some intuition about this. Let's, as an exercise, let's take a capacitor and say that it's connected to some circuit that I can't see, but I know something. I know that there is a constant voltage V over this capacitor. Okay? So the voltage over this capacitor is constant. What can I say about the current? Let's use the relationship between current and voltage for this capacitor. V.
Also constant.
What is the constant? No. V is a constant. V is equal to some V0 number, 5 volts. You have to differentiate.
Zero.
Yes. So I is 0. There is no current flowing. This current is 0. No current is flowing through this component. How come? Why does this make sense? Because if current is flowing, then charges accumulate on the plate. The amount of charge increases, and thus the voltage increases. But I just told you that the voltage is constant. So when the voltage is constant, I have some amount Q of charge that is already there, and nothing is flowing by. If anything would flow by, I will have a changing voltage. Whenever charges go, whenever charges flow, I have changing voltage. When they stop, the voltage stays constant. Okay? So now let's do the same with constant current. Now I don't know the voltage, I'm asking you, but I know that I'm forcing constant current through this capacitor. So what happens to the voltage? Let's try to solve for the voltage. So dV over dt is equal to I over C. Do you agree? Yes. And I is a constant. So integral over this, dt. It's an integral over a constant. What is this integral? Yes, it's the constant times t plus some other constant. I'm using a because c is the capacitance. Okay, because we have a family of solutions for this, right? Just integrate it over a constant. So what did we get? We got that v is a linear function of time. What does it look like? This. It will go to infinity as time goes to infinity, right? Now, this makes sense because I force a constant current. I always supply 1 coulomb per second, for example, if it's 1 ampere. 1 coulomb per second. So every second, 1 coulomb is added to each one of the plates, such that the voltage grows linearly. Okay? Now, of course, this can never happen in real life. It doesn't have anything to do with reality because at some point there will be too much charge and too high voltage such that the capacitor will burn. Okay? And this material will be conducting and everything will go by it. Okay? It wouldn't be a capacitor anymore. So this is not something you do. If you have a capacitor, don't enforce constant current to it. The current has to go to zero at some point.
Explain again, how did you make that from the cube to the t, the c times dt?
Yes, I use this. Okay? I know that if I have a capacitor with a capacitance C, and it has a voltage enforced over it, V, then it means that the charge on the plate is Q. And vice versa. If I have a capacitor C and I know that there is a charge Q on its plate, then the voltage over it is V. And this formula holds. Now, I can use this because I know, on the other hand, that there is a relationship between current and charge. The change over time in the charge is the current. Okay, so all I did was to take, this is the derivative of Q over time. So the derivative of Q over time is the derivative of this side of the equation over time. This is the constant, and this is the derivative of this. Okay. By the way, both of these very simple components are linear. Okay? They are linear if they are ideal, of course, which is never the case. Then they are linear components, and the relationships between I and V, even though we have a derivative there, the relationships are linear. Okay, so I hope you understood a little better what is this capacitor. And in order for us to construct our first RC circuit, then we also have to know sources of voltage and current. Can I get a watch from someone? You don't need it? Or no? Yeah. Thank you. Sources of voltage and current. Because if I just connect two components to one another and close the circuit, most likely, well, something might happen depending on how much charge we have on the plate of the capacitor, but it's more interesting if I can supply and decide how much current or how much voltage I want the circuit to have, some part of the circuit. So what we're going to use is two types of sources. So unlike these two components, which are passive because they don't supply anything, they just react to what you force on them, the next three components are active. They supply current or voltage. Okay, so it's very simple. It's a voltage source. And a current source. So I'm just going to give you the symbol. Voltage source is usually, that's usually a symbol, where the longer plate is for the plus and the shorter plate is for the minus. The current source is usually just a circle with an arrow. The arrow indicates the direction of the current. So for example, a voltage source, I can buy in the store a voltage source that supplies 5 volts constant. It's a battery. Okay? Example, V is equal to 5 volts constant. Or V can be a function of time. Okay? So time. Okay? And the same here. I that is supplied by this source can be some constant, 1 ampere, or any function of time, depending on what you now, let's talk a little bit about directions, okay? Because we have 2 active components, 2 passive components. So I'm going to draw resistor and capacitor. And just for you to not make too many mistakes with signs, plus and minus, I want to just give you one time all you need to know about directions in order. And if you follow this, then there will be no mistakes. Let's say that I decide to mark the current through the resistor in this direction. I'm always referring to positive charges that are flowing. Okay? Even though we know that many times electrons are the ones that are flowing and they're negatively charged, we are treating them as positively charged going to the other direction. Okay? It's identical. So I'm always talking about positive charges that flow. And let's say that I decided that the flow here is from left to right. So where is the higher voltage? Here or here? Left, right. Okay? Or in an arrow, this direction. Okay? So for a passive component, I and V are always in opposite directions because I'm always referring to positive charges that flow. Also in the capacitor. The capacitor, if I decide that the current is flowing to the left, then the voltage is the current is flowing to the right, then the voltage is pointing to the left. Okay? On the contrary, for active components, the direction of the voltage and the current are the same direction. Why is that? Because for passive components, we know that if the current is flowing to the right, then it's running away from the positive. Okay? Current is always running away from the positive. Here, current is originated in the positive. It's coming from there, and there is some chemical reaction that causes it to be generated. Okay? Generated. So here, this is the direction of the voltage, of course, because I just told you that the longer plate is the plus. So this is the direction of the voltage. There's no argue about this. And this is the direction of the current. Same direction, both of them to the left. Also for current, this is the direction of the voltage, and this is the direction of the current. Now, what is the relationship between V and I for these sources? We said that this is the most important thing that we have to figure when we talk about components, the relationship between I and V. But for some reason, I don't talk about it here. Why is that? Because we're dealing with ideal components. For ideal components, the voltage is always what the manufacturer said, no matter what the current is. For any current that you force through this, you'll get the same voltage. Okay? No linear relationship like here. Also here, the current will always be 1 ampere, no matter what voltage you force over the size of the component, the terminal. Of course, it's not the case in real life, but for ideal components, this is true. Okay. So now we know all we have to know in order to build our first circuit and try to analyze it. sure that each one of the components has its two terminals connected to something. Okay, none of them is hanging in the air. Okay, so I have a resistor R with a resistance R, a capacitor with a capacitance C, and I have a voltage source. Okay, let's say that the voltage source supplies some V of t, function of time. Okay, could be constant or sine wave or whatever you like. Okay? Whatever you find in store. So the first thing you need to do when you analyze, what do I mean by analyzing a circuit? By analyzing a circuit, I mean that if you know, for example, V of t, and I'm asking you, okay, tell me what is the current through this branch, or tell me what is the voltage over this resistor as a function of time. I want you to give me all the voltages and all the possible voltages and currents in this circuit. And if you're able to do this, then you've analyzed the circuit. Okay, this is what we're doing now. So the first thing you need to do when you analyze a circuit is mark your unknowns, your knowns and unknowns. Basically, your unknowns are all the voltages and all the currents through all the components. Okay? So I'm going to start drawing arrows and give them names. The current of the resistor will be IR. The voltage of the capacitor will be VC. Okay, so the important thing to know here is about direction. So I can decide that the current goes in this direction. But once I've decided this, then I have to be consistent with myself and mark the voltages accordingly. Okay, so let's say that this is the current through the resistor. So this is the voltage through the resistor, and I'm going to mark it VR. Okay. Now let's mark the current through the capacitor in the same direction, IC. So this would be the voltage through the capacitor. Now I also have the general current. This is the voltage over the voltage source, and I also have a current through the voltage source. I will just call it I. This is the general current. So I have I, IR, and IC, V, VR, and VC. 6 variables. I assume that V of t is known, otherwise I cannot solve. Okay, so I have 5 unknowns. I, IR, IC, VR, VC. Yes?
Is the I on the bottom in the other direction? So this goes facing the...
Sorry? What?
The I next to VT, should it be the other way?
Well, I'm just looking at locally at the components right now. And for the components, we know that for active components, the voltage and the current are in the same direction. You're right that if you already noticed that this current has to be equal to this current, and this is correct, then they will be of negative signs. Okay? But it's just a matter of marking. Okay? So just locally keep the... I could have done everything here the other way around, but I just have to be consistent per component. Okay, so we'll see. Yes. Okay, so the first step is to mark. And the second step, in order to be able to solve, then we're going to need 2 rules. For example, your name is Lily? Yes. Lily noticed that there is a relationship, a very tight relationship between IC and I, and probably between IR and IC. So let's try to formulate the rules, the electrical rules, that allow us to understand this relationship. Okay, so we have 2 rules. I'm going to give them, and then we're going to take a break. Sorry. The rules are laws of Kirchhoff. Double H. Kirchhoff. The current law. And Kirchhoff's voltage law. And we never write all this, we just write KCL sum of the currents flowing into the node is equal to the sum of the currents flowing out of the node. For any node in the circuit, a node is just any point that you select in the circuit. The sum of the currents flowing into the node is equal to the sum of the currents flowing out of it. Okay, and the voltage law, we're going to discuss it after the break, of course, says that if you go in a loop and sum all the voltages that you see on your wave in the right direction, then eventually you get to the same point where you started, you get 0. Okay, so let's look at it. The directed sum of the voltages around any closed loop is 0. You can copy it and we'll take a break, and I'm starting at 11. Please be back.
רק לציין שהקטע של השיעור הזה הרבה יותר סביר מהקודם.
כן? זה
לא,
מאוד אישי, אני תלוי גם...
זה קצת דימוי כאילו...
מספיק שונים.
כן, לא, האמת שאם זה פשוט שאני פשוט זוכר לפחות
אולי
מכמה...
מכיר.
אני לא, גם זוכר ששיעור כזה יותר לקחתי עד לחצי, כאילו עשיתי מתח אחד לפני שנים, אבל כאילו באחד עם קבוצה מסוים כאילו מאוד ליבדתי את זה, כאן זה כאילו זה יותר קל להיזכר באלקטרוניקה מהתיכון, אני מניח.
זה יהיה שונה מאוד משיעור לשיעור.
כן, לא, אני יודע שאת הזה של היולר אני מניח שזה יהיה כאילו... לא, זה פשוט שהשיעור שעבר באמת...
זה נורא קטן.
כן, לא, באמת כאילו פשוט, לא רק גם שוב לזה שהשיעור שעבר זה פשוט באמת היה ריצה, כאילו יכול...
כן?
כאילו יכול שזה משתמע להביא יותר באמת קצת חומר ברמה של כמו שעושים במתח, שהנה לשיעור של אימא עוברת בדיוק של החומר, שאת זה כאילו לכסות והנה דוגמאות אחד, כי זה כאילו באמת
מה, לתת כאילו לחלק לאנשים את הסיכום של השיעור?
לא יודע את הסיכום, וגם כאילו באמת כמו זה שפשוט את זה של הדברים שכדאי להכיר זה באמת בסוף שפסקתם, אבל זה שזה מפורסם אחרי שכבר מנסים לשחות בזה, זה בעייתי.
מה זה הדברים שצריך להכיר?
של זה שם בעמוד, זה של ה-recommended subjects to know, remember.
אין שם משמעות לפרסונליות.
כן, לא, אני יודע, אבל זה סתם כאילו בעניין שזה כאילו עוזר,
זה מי שרוצה לדעת מה יש, צריך לעבור לפיזיקה.
כן, פשוט שהתשובה לה זה כבר היה שכבר נמצאים בתוך העניין, שזה כאילו קשה פשוט להתחיל לקשת גם את זה וגם את זה, במיוחד שזה נושאים ש...
זהו, זה בעיה קצת שיש חומר הכנה, אנשים מה-ACC מקבלים מייל בספטמבר על זה, אנשים שלא מה-ACC אז הם לא יודעים על זה.
כן, כי אני באמת חיפשתי לפני, פחדתי שזה יהיה משהו כזה, באמת זה כזה לא...
כן, אולי צריך איכשהו להכניס את זה לשנתון. אני חושבת על זה באיזשהו גורם שהוא...
גם, כאילו, וגם זה באמת שכאילו השיעור, כאילו אני מבין שלא כאילו איך להצליב בו, אבל באמת לשיעור הראשון זה היה יכול להשתמש לו עוד שעה, שעתיים על
כן, אני כאילו סיימתי את החומר ועשיתי עוד מלא תקבילים, בגלל קצת... אין לי יותר מה לעשות בנידון, אבל יכול להיות ש... כאילו, אם ככה, היית צריך לשאול אותי יותר. בגלל שאני לחזור, כי היה לנו מלא זמן.
האמת שאני כאילו כן, כאילו האמת שזה חלק מזה פשוט שלא הספקתי לרשום חלק, אני העדפתי ללכת אחרי זה ולשבת בספרייה לקרוא מהספר דבר. לא, כאילו גם האמת, כאילו כן, כאילו, זה שחלק מהתרגילים של שיעורי בית אני ארצה אחר כך לשאול, כי גם זה בדרך כלל סימן רע שהתשובה שלך שונה מהתשובה של וולף אמר לך.
בדרך כלל, כן. אז בסדר, אז תבוא לשעת קבלה עם זה, ככה שהם...
כאילו השעת הקבלה כאילו זה שעה קבועה, או
לא, צריך
כאילו...
לקבוע איתי. אני היום אחרי השיעור קבעתי כבר עם 2 אנשים, אתה מוכן...
כן, לא, אני חושב שכאילו קבעתי איתך וגם היה אמרת שלך באמת שיש עוד מישהו.
אתה עדיין?
כן.
כן, סבבה.
סבבה, תודה רבה.
תגיד פשוט לשים את התא של מה שכתוב באנליטיקה וזה, מה?
זה היום.
שבוע הבא גם אני במילואים, אז יש מצב לראות לדעת.
אני?
כן. נכון. שרפת את זה.
טוב, הייתי חייבת, כי אחרי שכתבתי This is
כן,
final,
לא,
כאילו leave me alone לכל המיילים הזוהמים, אז כאילו פתאום אני משנה.
זה גם, האמת שזה נראה לי כאילו, לפחות 50-50, כי יש הרבה אנשים שתמכו מהעניין.
אני יודעת,
כאילו
כן,
זה בא להם
תיארתי
בסוף
לעצמי,
שזה...
תיארתי לעצמי, כן.
יותר מדי, אפילו
מה שלא
דיברתי...
אהבתי זה שאני שולחת 2 מיילים, ואולי רואים רק את הראשון ולא את השני, ואז כאילו אנשים לא יגידו.
כן, כן. לא, בסדר. אני מצטער שאני...
לא, אין מה לעשות.
כן. האמת התלבטתי גם אם בכלל לעשות כאילו את ה...
רוצה לעשות? למה לא? איזה תואר עשית?
עשיתי קוגניציה ביולוגיה, אבל עשיתי שנה רק של מדעי המחשב. כאילו עשיתי לנהלת 1 ו-2 וזה פשוט היה די מזמן.
לכולם זה עובר זמן, הרעיון זה שזה תשב לרענן את זה.
כן, לא, אז עברתי על החומר. וכאילו והשיעורים האלה לא מוקלטים, אבל נכון עכשיו? ויש מצב להקליט שיעור ספציפי?
אם יש
דבר
בקשה
כזה?
ממש יש איזה שבוע שהשיטה הזאת לא תהיה שלנו, אז אם השיטה הזאת שלנו ויש לך בעיה ספציפית, אז אפשר... לא, פשוט בניתי.
איתה את מדברת?
כן. אני יודעת שמקליטים אותי, כי התחלתי מיקרופון.
הבנתי.
כן.
יש סיבה שלא? לא להקליט כאילו?
סתם
כאילו
אני חושבת
לחדש,
שזה
כאילו?
בכיתה הקטנה. זו פעם ראשונה שזה בכיתה גדולה.
וואלה. למה? יש שומעים כאילו שם?
לא, כי הכיתה הקטנה פתוחה. כל מיני פגישות של אנשים חשובים.
הבנתי.
יש מצב, לא נערכתי. אתה יודע, זה לא כל כך כיף. אבל זה לא סיבה מוצדקת, אז כאילו, אם
לא,
עכשיו תרצו אז נקליט, כאילו.
סתם כן אני אומר כזה, זה עושה לי את החיים קלים, אז תשב מול זה
בכל השיעורים.
לא, כאילו
ספציפית
ספציפית
השיעור
כאילו אני
שלנו.
פשוט במילואים כאילו, אז זה עושה לי את החיים קלים, כאילו
תשלח לי
ספציפית,
מייל ואני אנסה, אם זה חשוב לך תשלח לי מייל ואני אנסה להבין. לא נראה לי שזה חלק כבר, אמרתי.
אני מחליף את חזי, בגלל זה החיים שלי יהיו קלים
אוקיי. אז הדרכים שלי גם יהיו קלים, אני אגיד לך, תטפל בזה.
כן, אז אני אמור להיפגש עם חזי שיסביר לי מה עושים בדיוק.
אוקיי, אז אם אתה רוצה אפשר שאני...
כן, לא כל
רק
שבוע,
שאני
אני
אדע
אשמח,
מתי,
זה
כאילו?
נחמד לבוא לפה כאילו לשיעורים, אני לא
לא,
שבוע.
יש לזה הרבה יתרונות להקליט, אני לא...
כן. לא, זה
מאוד עוזר לאנשים.
כן, לא, זה...
כן, בסדר.
אבל שוב, ספציפית רק ל... לא צריך את כל השיעורים, ספציפית רק